everyone, welcome to the part 2 of photosynthesis. In the previous video, we started with the process of photosynthesis in brief, and we also started with the light reaction. In this video, we'll be studying the light reaction in detail. We already discussed the location of the light reaction, that's the thylakoids, its components, that's the photosystem 1, 2 and 2, and certain events of the light reaction, that's the light absorption, water splitting, oxygen release, formation of the energy intermediates. We have also given the brief of the light reaction through this diagram, that the photolysis of water releases the electrons into the photosystem 2, which are transferred through the electron carriers such as the plastoquinone, cytochrome B6F and the plastocyanin to the photosystem 1. From there, the photosystem 1 transfers the electrons to the ferredoxine iron sulfur complexes, and at the last to the NADP, forming the NADPH. During the photolysis of water, the protons were also released into the thylakoid lumen, and the plastoquinone cytochrome pump also releases certain protons into the lumen. These protons add to the proton gradient, which helps in the formation of ATP from the ATP synthase. Both the NADPH and the ATP are utilized in the dark reactions in the further, which we are going to study in the further presentations. First of all, we are going to discuss the Emerson effect. Emerson effect is the increase in the quantum yield or the rate of photosynthesis when the light of the shorter wavelength is provided element along with the higher wavelength of the lights. Let's discuss what happens in the Emerson effect. When we provide the monochromatic light to the plant, the photosynthetic rate drops down. But when we supply the plant with the supplementary light of shorter wavelengths, the increase in the photosynthetic rate is observed. You can see in this diagram, when the higher wavelength of the light, more than 680 nanometers, that is 710 nanometers of the wavelength of light is provided to the plants, the photosynthetic rate was quite low. You can also see the relative account of when the 650 nanometers of the light, that is shorter wavelength of the light, was provided, only the photosynthetic rate was comparatively higher. But when both the lights are provided in the supplementarily, means along with each other, when the 710 nanometer and the 650 nanometers of the light are provided together to the plant simultaneously in the turns, then the enhancement of the photosynthetic rate was observed. This is called as the Emerson effect. You can read the Emerson effect from this slide. Let's proceed forward. We'll be studying the photosystems now. The photosystem 2 consists of the trap center, that's P680. That means the photosystem 2 absorbs the light at 680 nanometers. From there the electrons were picked up by the pheophytin. So next component is pheophytin, then the plastoquinone, then the ubiquinones, then the cytochrome B6F, and at last the plastoquinone. The ubiquinone and the plastoquinone are the mobile carriers. Let's proceed forward towards the next slide. In this slide, you see the light harvesting complex. Light harvesting complex is actually the complex of chlorophyll A molecules which gather the light energy and transfers it to the reaction center. Let's see how this occurs. When the beam of the light in form of the proton hits the chlorophyll molecule, the light energy gets converted into the resonance energy. The resonance energy is transferred to the adjacent chlorophyll molecules in form of the vibrational energy. But as the chlorophyll molecules are fixed at their own place, uh, they can transfer the vibrational energy by the vibrating at their own position. That's a to and fro motions. I'll show you how it occurs. When the photon hits the chlorophyll molecules, the molecules convert the light energy into the resonance energy. The molecules start vibrating at their own place. In the same way, the vibrational energy reaches the reaction center. Here's the cursor. Yeah, and after it reaches the reaction center, the reaction at this time the reaction center has already accepted the electron from the electron donor. That in case of the photosystem 2, it was the electron evolving complex, or you can say the oxygen evolving complex. When it gets the resonance energy, the electron gets excited, and it is transferred to the higher energy orbitals. From there, the electrons are picked up by the photophosphorylation. Let's proceed forward to the photosystem 1. And photosystem 1 consists of the trap center, that's a P700. That means the absorption of the light takes place at 700 nanometers. The iron sulfur complexes, ferredoxin, plastoquinone, and cytochrome B6F. Let's proceed forward. This is the Z scheme showing the movement of the electrons in the photosystem 1 and the photosystem 2, which we have already discussed. Let's do a brief. The light hits the chlorophyll molecules, the vibrational energy reaches the reaction center, the electrons move to the higher energy orbitals, then they are transferred to picked up by the photophosphorylation, transferred to the ubiquinones. The ubiquinones does not move further until each molecule of the ubiquinone gets one electron. It is having two ubiquinone molecules, the QA and QB. After that, the electrons are transferred to the cytochrome B6F, that's the iron sulfur protein. After that, the electrons are transferred to the plastocyanin, that's another mobile carrier. The plastocyanin handovers the electrons to the photosystem 1. The photosystem 1 also gets another illumination of light, or we can see a beam of light. The vibrational energy again reaches the reaction center. Again, the molecules get excited to the higher energy orbitals. Then electrons from there are picked up by the iron sulfur complexes, then transferred to the ferredoxin, at last to the NADP, forming the NADPH. Let's proceed forward towards the difference between the photosystem 1 and photosystem 2. We have already discussed the photosystem 1 and 2 many times in this video. Let's do a brief about the differences. In photosystem 1 is located in the appressed part of grana. Photosystem 2 is located in the non-appressed part of grana and grana thylakoids and the stroma thylakoids. The chlorophyll to carotenoid ratio is high in photosystem 1 and it's low in photosystem 2. The chlorophyll A content is more than twice the chlorophyll B content in the photosystem 1. In the photosystem 2, the chlorophyll content is equal to the chlorophyll A content is equal to the chlorophyll B content. The photocenter is P700 in photosystem 1. It's P680 in photosystem 2. The photosystem 1 receives the electrons from the photosystem 2 and the photosystem 2 receives the electron from the photolysis of water. The photofossilization, in photosystem 1, the cyclic photofossilization is occurs, and in photosystem 2, the non-cyclic photofossilization occurs. Photosystem 1 handovers the electrons to NADP and photosystem 2 handovers the electron to photosystem 1. Let's proceed forward to photofossilization. What is photofossilization? It is the ATP formation in presence of light. We need an electron transport system for the ATP generation, of course, and along with that we also need the proton accumulation into the thylakoid lumen. That we've already discussed. The two proton pumps are responsible for the accumulation of protons into the thylakoid, that's the photolysis of water, and the cytochrome plastoquinone cytochrome pump. let's see here how much amount of protons are transferred by each proton pump. The photolysis of water resulted in the introdonation of the 4 protons into the thylakoid lumen, and the cytochrome plastoquinone cytochrome pump transfers the 12 protons into thylakoid lumen. This much amount of protons are enough to build up the proton gradient, which resulted into formation of the ATP. The proton, the transfer of the proton across the membrane is coupled with the ATP formation in this ATP synthesis. Here, the two protons moving out of the thylakoid membrane resulted into the formation of one ATP molecule. The ATP and NADPH produced in the light reactions will be used in the Calvin cycle. That's a part of dark reaction. Let's proceed forward towards the types of the photofossilization. The photofossilization is of 3 types: cyclic, non-cyclic, and pseudocyclic. Cyclic and non-cyclic we have already studied, and this photosystem 1 and 2. And the third type is pseudocyclic photofossilization. In the cyclic photofossilization, the electrons expelled from the oxidized chlorophyll molecules are never returned. The electrons expelled from the oxidized chlorophyll molecules return to the oxidized chlorophyll molecule by passing through a series of electron carriers. In the non-cyclic photofossilization, electrons are expelled out but never return to the oxidized chlorophyll molecule. Whenever it requires new electrons, they receive the electrons from the anionic decomposition of the water. In case of the pseudocyclic photofossilization, the oxygen-dependent photofossilization occurs by the FMN protein or vitamin. That the FMN breaks the water molecule to produce the FMNH2 and water, and ATP is formed simultaneously. It is coupled with it. Let's see how this occurs. First of all, we'll discuss the cyclic photofossilization. In this, you can see the electrons are enter the high energy orbital after getting the illumination from the sun rays. The electrons from here pass through a series of the electron carriers that carries out the oxidation reduction reaction, that are redox reactions. This means these electron carriers are reusable. After this, electrons are returned to the oxidized chlorophyll molecule. Here you can see certain redox reactions occurring during the cyclic photofossilization. Next, we'll study non-cyclic photofossilization. Here, the electrons which leave the oxidized chlorophyll molecules are never returned to them. They are transferred through the Z scheme to the NADP, forming the NADPH. And whenever it requires, the oxidized chlorophyll molecules requires new electrons. They are provided by the oxygen evolving complex.
Let's start with the pseudocyclic photofossilization. In this pseudocyclic photofossilization, the splitting of water occurs with the help of FMN. In this, the splitting of water is coupled with the ATP formation, and as this FMN is acting as a coenzyme which can be used again and again, cyclic process is formed in which splitting and the formation of water occurs in a cyclic way, and the splitting of water is coupled with the ATP formation. Let's proceed forward towards the photolysis of water. In photolysis of water, the protons are formed which are used in the proton gradient for the formation of water. For the ATP formation, electrons which are passed to the photosystems, and of course the waste products, oxygen as a waste product which is not required by the plant, so they are released into the air. Here you can see the oxygen evolving complex. oxygen evolving complex mainly consists of water molecule, manganese ions, calcium ions, and chloride ions. In presence of light, splitting of water occurs, releasing the electrons, which are picked up by the photosystems, and after the splitting of water, the half molecule of oxygen and protons are released. Let's proceed forward towards the oxygen evolving complex in detail. In the oxygen evolving complex, you can see here, as I have already told you, there are the manganese ions in the oxygen evolving complex. This is the water molecule in which oxygen is attached to the two protons. One proton is this side, and the one proton is present on the other side. During the, you can see here, the photolysis of water is taking place. The protons are released into the lumen, and the electron is released, which is being transferred to the reaction center of photosystem 2. You see here, reaction, we have the ATP and NADPH2 that are used in the dark reactions, and of course the protons used in the formation of the ATP and oxygen that is released into the air. Let's see the relationship between the photosystem 1 and 2, the photolysis of water and synthesis of ATP. Photosynthesis begins. The electrons are provided to the photosystem 2 by the splitting of water. When they enter the photosystem 2, get excited and pass to the photosystem 1 through a series of electron carriers by the known cyclic photophosphorylation. Here the electrons are transferred to the NADP, forming the NADPH. The protons are also released during the known cyclic photophosphorylation through the plastoquinone cytochrome pump, and during the photolysis of water that are used in the ATP formation. And the photosystem 1 also does the cyclic photophosphorylation by evolving the electrons and passing them through a series of electron carriers, and then the electrons are again returned to the photosystem 1. In case of the non-cyclic photophosphorylation, once the electrons released from the photosystem 2, they never return to the oxidized chlorophyll molecule. Whenever photosystem 2 needs new electrons, they are provided by the splitting of water. That's all for today. Please like, share and subscribe to this video, and please mention in the comments if you want any specific topic to be covered. Thank you.